Good afternoon, everyone, and welcome to our CCOVI Lecture here on April the 3rd. It's my pleasure to introduce our speaker today, Dr. Jeff Stuart. Jeff is one of our CCOVI Fellows here in the Institute, but he's also an Associate Professor in the Department of Biological Sciences in the Faculty of Math and Science here at Brock. Jeff has a BSc in Biology and a PhD in Zoology from the University of Guelph. His wine-related research focuses on the biological activities of resveratrol and other red wine-related polyphenols. And he also looks at the molecular mechanisms underlying longevity, and many of those relating back to the function of these polyphenolics. Today he'll be talking about the topic of his upcoming book that we're all anxiously awaiting its release on bioactive polyphenols from wine grapes.
Thank you, Debbie. So what I'm going to talk about today might actually be a little bit lighter on data. I spoke here about a year ago in a talk that was quite heavy on data. And since then, we've got more data, and yet I'm going to present a little bit less. I am going to focus more on, I'll give the supporting data, but I'm going to focus a little bit on the book, in the sense that the book is providing quite a broad overview of this area of research that includes our own results, but also many other people's. But I value the book. It was a project that Ellen, Rob, and I undertook because we felt there was a need to synthesize quite a bit of disparate data results into one story, a single story, because there's many. And so I'm not going to tell all the stories. I'm going to just tell one. And that one is the one that we emphasize in the book. So thanks for having me here today. One thing I'll say, too, is just interrupt me, if you like, as I'm going along. That's absolutely fine. It's not going to, I hope, negatively affect my concentration. We'll see about that. So this work is all really Ellen Rob's work in my lab. And she's a graduate student, a PhD candidate, and she's going to be upset that I point this out, but she'll actually defend her PhD. So you'll see all of the data in its raw form when she defends in about 3 weeks. So Ellen started in my lab, which the lab itself is the interests and the things that are being pursued are a bit broader and we're interested in the biology of longevity, especially the cell and molecular biology of longevity, broadly speaking. Ellen came into the lab and started to work with this molecule, resveratrol, that was at the time very much a hyped molecule in that field. Some data had come out saying that it promoted longevity in one after the other species, really. A lot of the typical biological models, Drosophila melanogaster, Caenorhabditis elegans, and so on. So invertebrates, people were building their way up to actually doing the mouse and other studies. So she came in to do an undergraduate thesis, and immediately we began to find some interesting things. And so I'll tell you a little bit about that today. It's a story that just kept on expanding and expanding, and into something that I see as kind of an opportunity for this particular region, because these molecules that I'm going to talk to you about are found in abundance in grape skin. And a lot of times the grape skin is basically a waste product of the process. If you're making juice, for sure, if you're making white wines, there's a lot of thrown away white grape skins that actually, in many cases, it depends, you'll see a little bit, on the conditions of growth, but under the appropriate conditions, they'll have a lot of these molecules that we're talking about. These are the molecules that I will talk about. We've got resveratrol, but we've also got these other molecules that are derivatives of resveratrol, and I'll spend some time focusing on them, because that's really the crux of the story as I see it. And then these two are derivatives of resveratrol, but there's many other such molecules. That's the book that this story ended up becoming. So basically our story fit into the broader context of the field. And it will come out, I guess, at the end of the month. And it's a book. It's a small book. So it's about 70 pages. And it's not written for a lay audience. It reads like a scientific review, but it's a review of these molecules. Now, there's a lot of bioactive polyphenols, a lot of molecules that have been studied in wine, and this is limited to still beans, to a particular family. So that's the story I'll tell. And basically, those molecules fall under a category, a family of molecules called phytoalexins. And these are molecules that plants make because they can't, in part, because they can't run away. Anchored in space, and when they have things that are bothering them, especially live things, so they're induced by both abiotic, not live, and biotic stresses, these phytoalexin molecules, but it's particularly relevant to the biotic ones, to live infectious organisms that negatively affect plants and that they're not able to get rid of in any way other than to produce molecules to slow their growth. So these are common in any vineyard, and they're common around here, and they stimulate the production of these molecules, quite a wide variety of molecules, including molecules like resveratrol and other still beans. So, for example, if you seed grapes with an infection, with a fungal infection, and you go and repeatedly sample the grape skin and other tissues, you'll find increasing the responses to increase the concentration markedly of those, many of those molecules. And I'm talking here sometimes about several orders of magnitude, so not just make about twice as much, but many, many, many times more. And resveratrol is produced that way. So resveratrol is one of those molecules. Here's the synthetic pathway. So all you need is phenylalanine and malonyl-CoA to synthesize trans-resveratrol. I'm going to call it just resveratrol. Trans-resveratrol is the full name. There is a cis-resveratrol that is actually not biologically active in people. So that's the synthetic pathway. Number of steps. One thing that's interesting is that all plants seem to be able to get this far. And only some plants can take it to, I hope you can see the bottom there from the back, only some plants can take it to that last step. And those are the plants that have this enzyme, the gene encoding still bean synthase. So still bean synthase, that first of all, that has provided an opportunity for people to produce transgenic plants that are not vitis vinifera, that are not grapevine, and any plant transfected with still bean synthase will be able to produce resveratrol. So it's being done in a variety of other plants. But what's interesting is that if you look at the genetic diversity in grapevine, they have many individual copies of still bean synthase genes. So they have a remarkable diversity of genes encoding that enzyme. And that's unusual. That's something that hasn't been observed in other species. So many species have no still bean synthase. A number of species have still bean synthase, have the gene, make the enzyme, produce resveratrol. But the unique thing about the grapevine is that it seems to have really proliferated that capacity at the genetic level. So maybe that means that that's a particularly important thing in that species. So they can produce resveratrol. And resveratrol, so we've been working with it just since about 2006, not very long. And even since then, we've seen it just go a little bit crazy in terms of the kind of attention that's been given to it. And also, it's making its way right out into the popular press and popular culture. So I took those, plagiarized probably, those from Google. I should not mention that, should I? From Google just yesterday. But these are just, if you Google resveratrol images, you will get hundreds, it looks like, hundreds of different companies that are they're selling it in some form or another. You can go down to GNC in the mall and you can buy resveratrol in capsule form. You can buy it as skin cream. It's out there. Nobody can pronounce it, but everybody wants to buy it. So there is already this major fascination with resveratrol. There's a whole body of literature, I'll show you in a second, whole body of literature that has to do with this molecule and all of the amazing things that it can do, some of which are no doubt true. But it sure is a big body of literature, and it does get complicated. But one observation immediately is that I showed you the pathway for synthesizing trans-resveratrol. And that's actually what I showed you was only a partial pathway. Trans-resveratrol is an intermediate. It is not the end of a biochemical pathway, but that pathway can continue in a number of different directions. Glycosylation of resveratrol gives you the posaid. Methoxylation gives you pterostilbene. And then there's quite a wide variety of oxidative oligomerizations that give you many, many different molecules. So an oligomerization just means to have multiple copies of resveratrol brought together, sometimes 2, sometimes 3 or 4. And it's an oxidative process, and it's probably not readily reversible. I tell you that based on my conversation with Jeffrey Atkinson, because I'm not sure I would know that, but he seems to. So these are reactions that he tells me are unlikely to be reversed readily if those were given to mammalian cells. They would likely be active in the form that they're in or not. So, that is an intermediate. And one thing I just want to point out is they're chemically modified, but there are some key similarities that remain. And the really important ones in terms of what I'll eventually tell you in terms of how it works is probably that hydroxyl, which is present in most cases. Certainly, I'm going to show you data for that, for pterostilbene, posaid, and trans-resveratrol, and that group is there in all cases. That seems to be the important ones in terms of its overall effect, and you'll see why eventually. So they're made, but they are also made in significant quantities. So in this is just a table I've taken from someone else's work. It's published in the Australian Journal of Grape and Wine Research. And what it shows is a simple experiment with grapevine, where someone has gone out to the field and they have infected intentionally grapevine with a fungus, P. viticola. So they've induced this infection. The plant is going to generate a response. They go back, they harvest grapes, and they're measuring the content of these molecules. Now, not detected at all. ND means not detected at day 0, so that's your control. DPI is days post-infection, so day 2 is 2 days post-infection. So not detected. Are they there? Yes, we know they're there. But whatever method they're using to measure it, it's below that detection limit. So those molecules are present, but as I say, very low, much lower levels. And by 6 days, when there's been quite a bit of time to generate a response, you see now quite substantial concentrations of these molecules. But what I wanted to point out is, here's trans-resveratrol, 6. Just above it is the posaid, and down here is pterostilbene. So 6 or 5.7, 2.76. It's not just that the other molecules are made, but generally speaking, they are at least as abundant as resveratrol. They're present sometimes, the posaid in particular, at much higher concentrations than resveratrol itself would be. So that's an important observation. And then if you look at all these long-winded names belong to resveratrol oligomers. So molecules produced by taking the backbone and combining several. Some of them are 2, some of them are more. And if you look at the concentrations of these, on average, they're in many cases even higher. And what I'll say is we can't get most of those molecules. We can buy, for our experiments, we can buy readily resveratrol, poseid, pterostilbene. Most of these, though, are not commercially available. They're just beginning to be. A couple of them are just coming to that point. People have them, but they're molecules that they're producing in their own lab. We would like to have them. We would like to be characterizing these, much as I'll show you that we've done for the other molecules. So, the way the literature has developed is that molecule resveratrol captured everybody's attention for these obvious reasons, publications saying that it promoted longevity. Most of those turned out not to be true. And some other connections as well. There's a lot of excitement and interest there. And so, lots of publications. And I did this yesterday, and it was about 5,500, so just on PubMed, which is going to capture most of these resveratrol related, but not all. It'll miss a lot of the agricultural journals. About 5,500 publications on resveratrol. These in PubMed would be the ones primarily on human health and related to that in some way. And if you compare it to these others, poseid, pterostilbene, and the viniferins, tiny amounts, around 100 of each of those. Now that's changing. So if you look at the 100 of each of those, it's building. A lot of those would be in the last couple of years. People are beginning to realize, I think, that there is potential in the other molecules, and that they're really maybe part of the same story, sort of part of the same cellular mechanism. But really what that represents, I think, is an opportunity. There's an opportunity there to understand the extent to which all of these things can generate the same effect. For example, the whole idea of purifying resveratrol from an extract that contains all of these things might not be time well spent if they're all able to give you very similar effects in a human biological context. We started to work on these molecules just within the past couple of years, and we're trying to consult the literature to find out everything we could about them. And we really did realize that the story was running in parallel with the resveratrol story in many aspects. So if you ask this question, do resveratrol's derivatives share its biological activities? And here I'm not talking about biological activities in plants, but in people. That's what we're working on, what we're interested in. And one of the most robust observations made with resveratrol in the book, there's a table of about 40 to 50 different experiments, and that is taken just from about a 5-year window, the 5-year most recent window. So many cell types, many kinds of, different ways of doing the measurement. Some in vivo measurements on cancerous cells. A robust observation is that it slows the growth of cells. And we think we know why it does that, which I won't present much of that data today, not in depth at least, but Ellen will present it later in the month. So certainly resveratrol is well known to do that. Question is, do the other molecules, do poseid, do pterostilbene, and to a lesser extent do the viniferins, these other oligomers, do the same thing. Now, for that last class, I can't, there's very little data. But certainly for the first two, the data that's out there is saying yes.
When you mentioned about the in vivo studies, are they injected to the animal or the mouse, or have been consumed, like have been going through the digestion?
Okay, that's a good question. So the question really is, how do you deliver resveratrol to an animal? Most of those experiments are done in, not surprisingly, mice and rats. And there's a variety of ways that the resveratrol is delivered. Sometimes a dietary supplement, so added straight to chow. In that case, it matters what you how you deliver it. It seems to work well in a fatty vehicle, a lipid vehicle. Other times oral gavage, other times injection. So there are a number of ways. So all of them do, in fact, do this. The Pisaid and pterostilbene studies are generally much more recent, and we do these measurements as well, and we see, in fact, pterostilbene is better than resveratrol in the sense that it'll elicit very similar effects on slowing the growth of cancerous cell lines in the lab, but at lower concentrations, appreciably lower concentrations, which is good because concentration can be a problem in vivo. I'll mention that a bit later. Cardiovascular effects, these were some of the first effects attributed to resveratrol, cardiovascular system, and recently there's been some, these are generally effects on blood pressure and blood vessel dilation, nitric oxide system, and some other effects. And a lot of these, the data that's out there suggests that they can certainly be replicated with either pterostilbene or Pisaid. There's a literature on neuroprotection. Some of that is probably because of neurovascular effects, and some of it is clearly not that, but effects on neurons themselves or supporting cells in the brain. And that literature for resveratrol is relatively large. It looks like pterostilbene and Pisaid are telling the same story. Metabolic disease, a little bit of data for that as well. But generally, you can see what I'm pounding, strongly emphasizing the fact that the data that's out there, it really is the story is quite strong, saying that these are all doing the same things at the level of a whole body. Most of it's modeled in rat and mouse. There's a limited amount of human data, and that has made its way into the book. And there are some human trials going on with resveratrol, and less so for the other molecules, that support the idea that there's similar effects to these rodent models. So those derivatives of resveratrol have very similar phenotypes in people. And one of the things we've done some in vivo work as well, but I'm going to just show you today our work done in cell culture. So we do a lot of work with cellular mechanisms. We're quite interested in exactly if these different molecules are eliciting the same types of effects. It suggests that they're probably interacting at the cellular level in a similar way. And if we can identify what that looks like, then that information will really help us to understand exactly how that process works and how it could possibly be manipulated. So that's Ellen's data. And one of the early observations we made with resveratrol was that, so when you buy resveratrol in a bottle, they'll often talk about what a wonderful antioxidant it is, and even have maybe quantified its antioxidant capacity in that particular preparation. And we would argue that it doesn't matter how good of a chemical antioxidant it is. It's present at such low concentrations in the body that the chemical antioxidant capacity is likely not to be relevant. So once it's gone through the GI tract and been absorbed and metabolized, there's so little left that the chemical antioxidant activity is likely not important, but its biological activity remains. And you'll see that we really are arguing that it is a hormone receptor response, and we know that those can be amplified. Small amounts of molecule can give you a big effect when you start to talk about signal transduction systems. So we don't think its chemical antioxidant activities are important, but one of the things that we noticed immediately is it has this ability to modulate the normal endogenous cellular antioxidant system. So every cell in your body produces all kinds of antioxidant enzymes, superoxide dismutases and catalases for getting rid of the free radicals that are produced during normal respiration, normal metabolism. And that's a big group of enzymes. And what we've noticed is that almost none of that big group of enzymes is actually particularly affected. So it's not like there's a broad induction of antioxidant enzyme capacity. It's actually just one that seems to be very robustly stimulated by resveratrol, and what I'm showing here is by these other molecules too. That one enzyme is manganese superoxide dismutase. It is an enzyme that's found in only one place, and that's in mitochondria. And mitochondria are the source of most of the reactive oxygen or free radicals that cells produce, most cells produce. And so having that manganese sod, superoxide dismutase, in that particular compartment, in that organelle, seems to be really important. And we know that from a lot of studies where manganese sod has been knocked out. It's not compatible with life in mammals. So it has this important role. It also has some it has some signaling functions, what appear to be redox signaling functions that are less well understood. Main point here, though, is this is a Western blot which measures the amount of a protein that's present, and these are all controls. These two are controls. These two we can ignore for now, but they're, well, I guess we won't ignore them. I'll tell you what they are. They're metabolites of resveratrol that are produced in your body. So when you've ingested resveratrol, it's converted into some other forms and excreted. And then these are the original molecules, resveratrol, pterostilbene, and psaid. So this one ability to upregulate that enzyme that we've already attached previously to some really interesting outcomes is shared certainly by pterostilbene and psaid. There seems to be very little difference in terms of how robustly that is, that response is. We know from other studies where we knock down or knock out manganese sod, that mitochondrial enzyme, that it's required to get this effect. So this effect comes in next. And what's being shown here is the population doubling time of the cells that are being exposed to these molecules. Population doubling time means how quickly the cells are growing. The slower they're growing, the higher will be those bars. It means it's taking more time for the population to double, to go from 50 to 100 cells. And so resveratrol slows cell growth. That's the observation there. In this context, the higher bar is meaning slow growth. That's longer to double. And what we see is, psaid and pterostilbene are doing the same thing. This is with one particular cell type. These are C2, C12 cells. They're a muscle progenitor cell. But we have shown the same thing with many cell types at this point. Neurons, fibroblasts, other myoblasts, from a variety of species, from humans, from mice, cancerous cell lines, normal primary cell lines. It's not discriminating against cancerous cells and normal cells. It slows the growth of cells through a mechanism that actually involves manganese sod. But the bottom line here is simply that all of those molecules, resveratrol, pterostilbene, psaid, are doing the same thing, giving you that same outcome. And the other, so that has relevance in terms of cancer and the observations made that resveratrol and these other molecules are slowing the growth of cancerous cells. But a lot of the observations made in people and in rodents have been about things like neuroprotection, cardiovascular health, and those types of things might relate more to this kind of phenomenon, which we also observe widely with different cell types, and that is that treatment of cells, and it seems to be, this is a general phenomenon, so many different types of cells, treatment with resveratrol, so this is a pre-treatment for several days, so it's a chronic introduction and maintenance of resveratrol levels, and you get this response, and the cells become stress resistant. In other words, they're resistant to dying. A variety of stresses that would normally kill cells will now not kill those cells when they've been pre-treated with resveratrol. So that's what's shown here. This is the LB50 for hydrogen peroxide, which is an oxidant that is lethal. So it's basically the amount of hydrogen peroxide that is required to cause cell death. And these are some controls. And this is with resveratrol. So in other words, pre-treatment with resveratrol will require that more hydrogen peroxide be present to be lethal. And here is Passaic and pterostilbene, and telling us that observation too of stress resistance is not specific to resveratrol, but is present for all of those related derivative molecules. So the story then is that Passaic and pterostilbene have very similar activities to resveratrol when in experiments that have been done in generally in mice and rats, intact, dietary administration, sometimes other modes of delivery, and what we consider to be some of the important effects ex vivo in cells growing in the lab. Again, we see very, very similar effects. So I'm going to just switch gears a little bit to answer this next question. How can these three molecules that are different give you these same effects that are actually fairly broad? There's a number of different things about how the cells will behave that change when they've been exposed to these molecules. And one way that could happen, of course, is if they all were triggering and stimulating the same signal transduction pathway. We know that the way hormonal signal transduction pathways work is that they start from a node and they spread out, and they can become more powerful, so amplitude can increase, and they can become more widespread, so the number of downstream targets can increase. And we think we know what that pathway is, and I'll show you the data that supports that. And a lot of this is, again, it's emphasizing what I'm going to show you is emphasizing the book, and so a lot of it is actually circumstantial data, and then there's some of our more controlled cell culture data. But the beginning point is here, that trans-resveratrol actually has some structural similarity to a molecule that we're more familiar with, 17-beta-estradiol, an estrogen. And again, it seems to be the hydroxyl that is probably particularly important. Early studies, so from 1997, demonstrated the ability of resveratrol to bind to estrogen receptors. And so that there is a demonstration of its estrogenic activities. But I think it's hard to ignore in the literature the overwhelming similarity between the kinds of effects that are associated with estrogen and those that have been demonstrated for resveratrol. A lot of these, again, are similar to what I was showing you before. So cardiovascular problems, neurovascular and neural issues, cancer, metabolic disease. There's certainly a role for estrogen in all of these. And you see it most prominently in either overreactomized mice and rats or postmenopausal women. Estrogen appears to provide some protection from a lot of these common ailments. And that can be reproduced very nicely by overreactomizing rodents. And the protection conferred by the endogenous estrogen goes away. And then you can reintroduce estrogen by, for example, injection, and you'll reacquire that endogenous protection or now maybe not endogenous, but that protection that was there. So that has been observed for many, in many different contexts, protective effects of estrogen. And if you line them up next to what has been observed with resveratrol, and it's interesting that a lot of the work with resveratrol has actually been done in male rats and mice. And you might ask why, and one reason why is it's hard to see anything in intact female young mice. Whereas males with lower circulating levels of estrogens, there's less there to mask potential effects. So I'll just tell you a very little bit about estrogen and its receptors and how those work. Classically, there are two estrogen receptors, alpha and beta, that are both working as transcription factors. Now there's some non-genomic actions of estrogens as well, which we'll ignore for now. And we don't actually know how our particular mechanism of working, except for I'll tell you that it's estrogen receptor beta. So how does that look? Well, normally it would be estradiol that is going to diffuse into the cell and bind to one of those and initiate the transcription of hundreds of different genes. So any gene with an estrogen response element will be turned on by that. I've left out a whole bunch of partner proteins and dimers and heterodimers that occur. It is complicated, but you can simplify the system. Luckily, we have tools to simplify that bigger system. And the two tools in particular are, there's enough interest in estrogens and estrogen receptors and those kinds of effects that a variety of pharmaceutical companies have made selective estrogen receptor modulators. So agonists that are specific for either ER alpha or ER beta. And one in particular, diaryl-propionitrile, DPN for short, is a strong and selective ER beta agonist. And it's interesting that we can reproduce all of the effects, without exception. We can reproduce all of those effects that I mentioned for resveratrol, piceid, and pterostilbene with a selective estrogen receptor beta agonist. If we do the exact same experiment with a selective ER alpha agonist, and there are several of those, we've used primarily PPT, we see nothing. And that suggests to us that what we see is working through ER beta. Now, to know that, what you would need to do is some more experiments. We can pharmacologically inhibit estrogen receptors, but unfortunately, under our particular experimental conditions, we're only able to do that with a broad spectrum estrogen receptor antagonist that affects both alpha and beta receptors. Now, when we do that, the effects of resveratrol, piceid, etc. go away. So pharmacologically, we can show that it's estrogen receptors, and we know that it happens with ER beta agonists. But the last thing that we've been able to do is establish cell lines from ER beta knockout mice. So these are mice that completely lack the ER beta, estrogen receptor beta gene. They have no ability to make that receptor. And then just repeat the experiment. So if it was not important, then we should still see the effects of these molecules, the same kinds of effects. So that's what was done here. It's very difficult to see, I know. It's a bit blurry, but these are normal mice, ER beta plus, so the homozygous normal. And these are not mice, these are cells. These are cell lines isolated from mice. We've done it in fibroblasts and in myoblasts. So those are normal mice, and these are from ER beta knockout mice. So they do not have the ER beta gene, they don't have that estrogen receptor. And what's being shown here is that manganese SOD, that mitochondrial protein that's being induced. And this is how it's normally induced. So now we have an extra group. This is DPN. So I'm just showing you that DPN, the ER beta selective agonist, gives you the same thing you get with these phytoestrogens. You might notice it seems to be a slightly more robust effect, and it is a far better agonist of ER beta than these phytoestrogens are, than these other molecules. But in any case, it's the same effect. And if you knock out ER beta, that induction of manganese sod either goes away completely or is certainly blunted. What about the other effects? The main effect, one of the main effects in terms of cancer and the utility of these molecules in that context is the ability to slow the rate of growth. Of course, you can prevent transformation and the incidence of cancers, or you can slow down the rate at which cancerous cells grow, and these are both desirable objectives. So this is specifically the rate at which those cells grow that we're talking about. And here's the kind of effects. This is doubling time. Longer doubling time means slower growth. Higher bars are slower. DPN can reproduce exactly that same effect as those other molecules. And DPN is quite well characterized as being anti-proliferative, whereas the estrogen receptor alpha agonists are pro-proliferative. If you knock out ER beta, certainly that goes away. The effect with DPN goes away. And the effect with these other molecules, resveratrol, piceid, or pterostilbene, either goes away completely or is blunted. And finally, that test of stress resistance, we see the same thing. Very, very different effects in the ER beta knockout context than in wild type. So wild type cells, we see this resistance to toxins. Again, that's hydrogen peroxide. So you need more of the same toxin to cause cell death. You take away ER beta and that's gone. So we have concluded that at least these effects that we're interested in, and possibly much more of the story, is being mediated through this one pathway. ER beta regulates the transcription of hundreds of genes, but we also think that that mitochondrial manganese sod is a particularly important one. And just one last little point. So why not, this take-home message really is why you should not ignore these other molecules that are present in grape skins, and therefore also in red wines in particular. And it has to do with bioavailability. What is bioavailability? And it's something that would be measured in people or rodents. And basically it is how rapidly some molecule that you've introduced into the animal has been metabolized and excreted. If it's the more quickly that happens, the less bioavailable it is. In other words, it won't be able to exert its effects if it's immediately and totally eliminated from the system. Product of liver metabolism and excretion in kidneys. Resveratrol has relatively low bioavailability. Your body is really good at taking resveratrol and making it into these metabolites that can be readily excreted. And it does that quickly. And so it requires significant concentrations of resveratrol, far more, by the way, than you would get in your regular bottle of, or glass, depending on how you drink, of red wine. So we're talking about major supplementation. Lots of resveratrol delivered on a constant daily basis to sort of overcome that problem. If we think of it as a problem, I mean, it's your system doing its job. But to overcome that problem to some extent, you need lots of it, and you need it regularly. Both piceid and pterostilbene seem to have better bioavailability. So they stick around longer. Once introduced into the body, ingested, for example, they actually are eliminated less quickly. That could be good. So we've got these molecules that seem to elicit very similar effects to resveratrol. They're certainly present in the same source that we're interested in here, grapes, and they are not readily eliminated, not as readily eliminated. So are they better than resveratrol? They might be. They certainly need to be explored that way, which is why I'm saying, should it be resveratrol that everybody is so interested in, or does it need to be wider than that? Does the net have to be cast wider? And I would say that based on all of the information that we have, is a very reasonable hypothesis that, in fact, they're all doing the same basic thing. Now, there's differences. There's going to be, and the bioavailability of the viniferins, the oligomers of resveratrol, are not worked out. So there's lots of work to be done, lots of information to be gathered about these kinds of things. But certainly, we've got Piceid and pterostilbene we know a little bit more about, and they're fitting right into that story, suggesting that at least it should be all of those molecules that are sold as supplements, and not just this resveratrol. So, just to summarize what I'm saying and what I have said, phytoalexins are these molecules that are produced, and some of them are produced always, but certainly their production is upregulated strongly by stress. And I was focusing on biotic stresses, but cold seems to be able to do that as well, and other stresses, UV light, excessive UV. They're made everywhere. They're present in woody plant tissues. They're present in the pulp, but they're also present in the skin. And they're fairly abundant in skin. And when wine is made under fermentation conditions where the skin is still present, what I didn't mention is they're highly hydrophobic, and they'll leach out of that skin and into the forming alcohol. And those molecules are all phytoalexins. They're all produced in response to stress, fungal stress. For example, the downy mildew that's common around here will do that. They're all made under the same conditions. They're present in similar concentrations. It is somewhat strain dependent or cultivar dependent what the relative amounts will be, but they're all certainly abundant. And the literature, which is vast on resveratrol and smaller for these other molecules, seems to be very much a parallel literature. Quite similar findings in terms of effects on human health, or at least in the models we use for human health. And when we do the experiments in our lab, Ellen's experiments with these same molecules, we certainly can reproduce all of the same effects with Piceid and pterostilbene that we see with resveratrol, and in all cases they seem to be at least partly dependent on the presence of estrogen receptor beta. And we can certainly replicate the effects with an ER beta specific agonist. And I would note too that other ER beta agonists are being developed for treating a variety of symptoms in people. The leading one seems to be postmenopausal symptoms, MF101, and some are actually phytoestrogen formulations. So there's already a strategy in place there and clinical trials for these things that seem to be at least partially successful. And so the idea of targeting ER beta is out there, and it looks like these are weak agonists of ER beta, but they're strong enough to generate some effects. And I show you this table again, just to re-emphasize the point that out of that whole collection of molecules, we only have data for 3. There's very, very little data on human effects, human health effects, effects at the cellular level for all of these other molecules. And I think that's what we really could use at this point. And that's kind of why we wrote that book. It really is it's a sort of a big scientific review of that field, and that's the point we were trying to make, is that all of these molecules seem to be important. They seem to give similar effects and have very similar biological mechanisms at the cellular and molecular level, and they deserve our attention. So there's a number of things that I think need to be done that are relevant to, for this talk, relevant for the industry around here. We're talking about grape skins that are garbage in many cases, that are waste products, and have, I think, often essentially no value, and being able to capture some of that value. So what needs to be done? A better and more complete characterization of what these phytoalexins are doing beyond just resveratrol. You saw how biased everything is towards resveratrol. It's my opinion that that's not justified. So purified individual still beans, and then even combinations. We know that sometimes the matrix of delivery seems to be very important. Fatty delivery systems, delivery in fat, seems to be ideal. They're very hydrophobic molecules. It's no doubt working. It's because of that on some level, but we don't understand why exactly. Ex vivo, we've done lots of work with cells in dishes, but it needs to be moved into an in vivo context. It needs to be done at least in rodents to some of these, some more work. And one thing I didn't talk about much today, but is in, it is a chapter in that book, or part of a chapter in the book, is basically the relationship between growing conditions, but also storing of picked grapes. The conditions of storage are affecting the levels of some of these still beans in grape skin. So knowing how that works, there's people are working at that level. I would say there's not a lot of data there, and there should be more. So some attention to how to maximize those levels. Obviously, one way that's straightforward is organic growth practices that don't spray these grapes to solve the problem for the grape, for the grapevine, but allow them to solve it their own way, which is to produce these molecules. And I think this is the opportunity here, is there is a potential to, knowing those things and maximizing them, there's a potential to capture some value from those waste grape skins. So, thank you for your attention. Just to acknowledge the different organizations that have supported this, and mainly NSERC, an NSERC operating grant, and one of the Poseid and pterostilbene work was largely supported by a BUAF, a small grant from the university. Ellen's supported by NSERC's Vanier Scholarship, and all of the infrastructure from NSERC and the Ontario government. And thank you very much. There's slides.
I have two questions. First of all, the ratio of the cis resveratrol compared to the trans resveratrol, and also if the cis resveratrol would end up being the cis, those two molecules are.
So the ratio of cis to trans is generally lower, and the cis resveratrol becomes cis whatever else derivatives, and yes, the trans is the more abundant isomer, stereoisomer, but it also degrades to cis, so it depends on conditions as well. The cis, early on we did, and other people have done studies with cis resveratrol, and it doesn't seem to have any of those biological activities. It seems to be quite specific to trans resveratrol.
By itself, or could be acting as a synergy, but can it have a synergistic effect on the other activities of the other resveratrol or the polymers?
We don't know. We've done a very, very small amount of work where we've added these things together. It's a good idea, and that's one of the things that we'd like to do more of. And we'd like to actually, rather than start from the purified ones and adding them back, even do some work with just making some extracts from grape skins, characterizing them, and using those extracts directly. I think we need to do it both ways, but so we don't know about synergies, we don't know about interactions as much. We don't even really know enough about all of the individual molecules. We know just a small subset of those. Al?
I'm going to be a Grinch for a bit here. I was wondering about any possible negative effects from these molecules.
Yeah.
And they're from three perspectives. One is that phytoalexins in general are produced because they're toxic to fungi. And also resveratrol is structurally similar to diethylstilbestrol, which is used as frowned upon now. And the third thing is when you showed the change in the LD50 for the hydrogen peroxide, I was wondering if that would have the effect of inhibiting apoptosis in cancer cells.
Yeah.
So in general, are there negative effects?
Yeah. I'm definitely, all of those points I think are true, although I may have forgotten them already. The first point in terms of, or maybe your second point, phytoestrogens being frowned upon, there's good reason for that. I didn't go into this story much, but resveratrol will bind both ER alpha and ER beta. Now we can see that our effects that we're seeing, that we're interested in, are definitely mediated by ER beta. We leave ER alpha in there, ER beta is gone, the effects go away. But it doesn't mean that there aren't other things that do go on with ER alpha. If you give estrogen to any cell and you see an effect, you in some cell types will see increased growth rate, and in other cell types reduced growth rate with estrogen, with 17-beta estradiol, for example. And what seems to be going on is that they have it's the ratio of ER alpha to beta. If they're primarily expressing the alpha isomer, they will respond to estrogens by proliferating. If it's primarily beta, which is the anti-proliferative pathway, they'll respond by slowing. So postmenopausal women, an estrogen replacement therapy was stopped. Those trials were stopped because of problems that were related to that broadness of response. The ER beta selective approach is being tested as a way around that. So they're only as good as they are selective, I think. And in terms of growth and development, so I think one of your other questions was, yes, okay, so cancerous cells and slowing them down but also making them more robust, that's true. You slow them down, but if you do the LD50 experiment in cancerous cells, they're harder to kill as well. Yeah. So it depends really on what you're trying to achieve. And I think people, when they're taking resveratrol, because people are doing that now. I'm not one of them. I don't think that there's enough known about it yet, but people do. Clearly, they're buying it. And they would be getting some sort of, if anything, because you need to take a lot, they would be getting maybe some broad spectrum slowing of cell proliferative rates. And yes, that could have some positive effect on cancers, but if you were then going to do chemotherapy or radiation therapy, you definitely wouldn't want that. You would, genistein is another phytoalexin or phytoestrogen that has been explored in the same kind of context. And in addition to all of those kinds of problems, there's another kind of problem that would occur during growth and development. So the rodent studies, certainly, with genistein say that you really shouldn't be exposing developing mammals to high amounts of those molecules because they will interfere with normal growth and development. Whether the same thing could be true of these other molecules, I'm not aware of there being much data on that, but the potential is definitely there. So they're not, so, I mean, that's kind of so in terms of encountering them in just normal grapes or in wine, which a 3-year-old shouldn't be drinking arguably anyway, we're not talking about, the levels are very low. But if you're supplementing with these, with what you bought at GNC, then you could actually get levels high enough that I think there would be that possibility. You would never want to do that in a non-adult.
Yeah. So, do we have any more questions?
It's along the same lines as what Dr. Gass was talking about. I mean, I thought it was a little funny at first, but Is there any serious behavioral or morphological changes that may occur as a result of taking high levels of resveratrol as an adult over time?
Yeah. So I'm going to answer your question in a slightly different way.
Sure.
The ER beta agonists, which are better, so much lower levels of those molecules will give you a much fuller stimulation of that ER beta pathway. Those are actually, research into those have been supported by the American military. And the idea is that they're anti-anxiolytic, which means they reduce anxious symptoms. And so I think, well, this is for maybe soldiers who are, for good reason, anxious, and taking, so some research has been done giving those individuals DPN. It clearly has effects. ER beta is somewhat ubiquitously expressed. It's certainly widespread, that receptor in cells of the body. And it's present in many parts of the brain at relatively high levels in males and females. And it clearly, the ER beta specific agonists clearly do have effects on anxiety and depressive behavior and some other phenomenon that would be clearly important. The question comes back again to, could you get enough of those molecules in, and the ER beta specific? So in our experiments, certainly these effects that we're following are dependent on ER beta. We're not seeing ER alpha linked effects on those exact same processes, but the molecules are potentially sloppy enough to be having those other kinds of effects. So again, it would need to be worked out.
Sorry, I'm asking a lot of questions.
That's okay.
Just something for non-adults things in 12. There was one study 2 years ago that they incorporated resveratrol into milk. So for the non-adult who cannot drink wine, they can use resveratrol in that case. But my other question, which is a lot broad in general, would you recommend with all of that, for example, fatty carriers of the resveratrol, would you recommend, for example, people who have fatty food with one?
Well, so there's a pretty extensive literature on resveratrol's ability to either prevent or lessen the negative effects of a high-fat diet, starting in rodents in 2006, and then more recently in people. So, yes, there is evidence there, certainly, that there are some benefits to taking resveratrol supplements, and I'm talking again much higher levels of resveratrol than you would get in wine, at the same time as you're having this high-fat diet and getting some protection, some beneficial effect. There's at least dozens of papers on that at this point. We've talked about that and again in that book. Not that I'm trying to sell everybody that book, because I don't really care that much, but it is, well, I mean, I care in one aspect, which is that we did the work to put it all together, and I think you can find, that'll link you up to all the references at least at this point, because it's fresh. Those are all the references should be quite up to date for now.
Well, if there's no more questions, please join me in thanking Dr. Stuart for stimulating There's a token of our appreciation, a wonderful Brock University book and a cutting pen. And last but not least, I just want to remind you that our last lecture in the lecture series is on Wednesday, April 10th. It'll be given by Dr. Jim Wilworth, and the title is Getting Through the Winter, Updates on Freeze Protection and Cold Hardiness